Okay, now to just further illustrate the importance of protein structure and protein binding to ligands, we'll talk about another example. And this is antibodies binding to their antigens. So first of all, just to recap, we talked about hemoglobin and myoglobin last time, and of course, again, they're the particular shape of the protein and the ability of the protein to bind to its ligand varies with shape. But actually, in a way, hemoglobin and myoglobin are a little unusual in that the binding of the ligand being oxygen, is not really a direct interaction with the protein. So it involves that prosthetic group, the heme group. But for most proteins that bind to their ligand, the interaction is directly with the amino acids that line the binding site. And that's important because, again, one thing that's important about protein ligand interaction is that there has to be some specificity. The protein has to bind specifically to its ligand and not to other maybe similarly shaped ligands. So we saw that a little bit with hemoglobin and myoglobin and carbon monoxide. So again, those molecules need to specifically bind to oxygen and not carbon monoxide, and the shape of the protein does facilitate that. But for bigger, more complex molecules, there needs to be even more specificity. So antibodies are a great example of that. So antibodies, if you know anything about the immune system, are created by specialized cells in your immune system. The cells are called B cells, and they have this very particular structure. So each antibody protein consists of 4 subunits. So you have 2 longer chains called the heavy chains and 2 shorter chains called the light chains. So it's actually a tetramer, but they are connected to each other. So the 2 heavy chains you can see over here are connected by a disulfide bridge, and then each light chain is connected to one of the heavy chains by a disulfide bridge also. So this is not how hemoglobin works. So the hemoglobin subunits, for example, are not connected by disulfide bridges. They're only connected by non-covalent interactions. And so within each of those chains, there are 2 types of domains. The heavy chains each have 3 what are called constant domains, and so they're designated over here C. And in fact, the 2 parts of the heavy chain that are connected to each other and form kind of the tail of this, or the stem of this Y-shaped arrangement, is called the FC domain. And the C again stands for constant. What that means is that for all versions of this antibody, but there are multiple types of antibody proteins, immunoglobulin G, or just IgG as it's abbreviated, is the most common type in your blood most of the time. So IgG proteins always have this constant domain that's always the same sequence. So every version, every copy of an IgG protein will have the exact same constant domain, including this FC domain. But then both the light and heavy chains contain these variable domains designated V. So VH means the variable domain of the H, or the heavy chain, and then VL is the variable domain of the light chain. And the variable domains are next to each other in the protein. They together with the parts of the constant domain create what's called the FAB part of the antibody. And the AB stands for antigen binding site. So the word antigen is just a special term for the thing that the antibody binds to. So it's the ligand of the antibody. Usually the antigen is something foreign, so some molecule that is perhaps part of a virus or part of a bacterial cell or some other parasite. But in principle, anything can be an antigen. Usually bigger molecules. So usually antibodies are not formed against or small molecules that might just be normal intermediates, common intermediates in the tissues, but larger structures tend to act as antigens. And the variable domain is variable because every copy of an IgG protein will have a different domain. Now, this is not the norm for proteins. Most proteins have a specific primary structure that is the same for that protein every time it gets made. But antibodies have a special way of getting made. They're transcribed and translated mostly the same way that other proteins are, except that those variable domains, before they're transcribed in the B cell, each B cell goes through a genetic recombination process where basically the DNA that corresponds to those variable domains gets chopped up and rearranged effectively at random. And so what that means is that every B cell makes a slightly different version of immunoglobulin G, and that means that each one has a slightly different binding site. And in fact, there are about 10 million different combinations that can be made, which means that at any given time, there are about 10 million different types of antibodies floating around in your blood. And each of those antibodies has the ability to bind to a different antigen. So the idea being that any potential virus or bacteria that enters your system will be recognized, or part of those proteins will be recognized by at least one antibody. And so when the antibody and the antigen bind, they form the antigen-antibody complex. So each antibody has two antigen binding sites, and that means that each antibody can bind to two antigens. Again, the antigen can be anything, so it could be part of a virus, or it could just be a virus or a protein or a toxin from a bacteria. But whatever it is, it will have a very high affinity for that antigen binding site. And again, that's not because not the way most protein ligand interactions work. So, if we're talking about a receptor, for example, the binding site on a receptor for its ligand is the way it is because over many years of generations of evolution, the protein sequence for that protein, the amino acid sequence for that protein, has been modified to make a protein that's precisely shaped for its ligand. But here, essentially, the antibodies are just picking random shapes, and with the idea being that any given antigen that shows up will bind to at least one of these proteins. Now, without getting into too much detail about how the immune system works, the way you develop immunity through antibodies is once these antibodies are bound to their antigen, one of the things that can happen is they can be consumed or by cells called macrophages. So a macrophage is this big kind of amorphous cell that moves around your blood and your tissues, and it's covered in these proteins called Fc receptors. So the Fc receptors, again, this is another example of protein ligand binding. The ligand for this receptor is the Fc region of the immunoglobulin protein. So when a macrophage encounters a antibody that has an antigen bound, it will stick to it. And if multiple of these Fc receptors are activated at the same time, that means that multiple antibodies are attached to one antigen. So in this case, it would be a virus. Now, these could all be the same antibody, or it could be different antibodies binding to different parts of the same virus. But regardless, all those Fc receptors being activated simultaneously trigger the macrophage to go through a process called phagocytosis. So this is a word that basically means the word phago means to eat, and cytosis means basically movement of the cell. So it's a process in which the cell kind of engulfs the whatever the antibodies are attached to. So if it's a virus or bacteria, then the macrophage just eats it, essentially. It destroys it. And so that's one way that the immune system can get rid of invaders, foreign invaders, and infections. But what can also happen is then the macrophage will take the protein or the antibody that it used to recognize the antigen, and it will express it on its surface, and that then triggers a whole nother set of processes by which the original B cell that made that antibody will proliferate and start making more copies of that particular antibody. So that means that the next time this virus shows up, there will be more of those antibodies present to take out that virus. So that's how you develop long-term immunity. It's also how vaccines work. So if you take a vaccine for, let's say, the flu virus, then you're basically being injected with a part of the flu virus particle. So that means that the virus, even though it's not infectious, still presents itself to the immune system as if you were infected. And so your immune system then starts making antibodies to the virus in response. But the important thing as again, it's the interaction between the variable domain of the antibody and in this case the virus protein, and then the that allow the antibody to bind to the protein, and then the Fc region of the antibody binding to the Fc receptor on the macrophage, which triggers the phagocytosis response. Again, it's important that the shape of the protein be ideal for binding to the antigen, and you even see examples of induced fit. So when the antibody binds to the antigen, the variable regions actually can come close together. You can kind of stabilize the antigen antibody complex by induced fit as well. Antibodies are really important for the immune system. It turns out they're also really important in studying biological molecules. So antibodies are used a lot in research for lots of different methods. What you can do if you want to study a molecule is you can make an antibody against that molecule. So if, for example, you want to study a human protein, you can extract that protein or purify it however you want to, and then inject it into another animal, an animal of another species. So often this is done with mice or rabbits, sometimes larger animals like horses or goats. And the host's immune system, because you're injecting it with a human protein, will recognize it as foreign, and so it will start making antibodies against that molecule. And then you draw blood from that animal, purify the proteins, and there you have an antibody that's specific for your protein of interest. And you can do this with lots of pretty much any type of molecule you want to. And then you can use that antibody to locate or study the protein of interest in other samples. So this is an example where you could take a culture dish and coat it with antigens from your sample. So let's say you want to know if my protein of interest present in a mixture of other proteins. So this is our protein of interest. So what you can do is put it in a culture dish and coat that surface with those molecules. So it'll be mixed with a whole bunch of other molecules that maybe you don't care about. Usually these reactions involve some sort of blocking agent. So the blocking agent is just something that's going to cover the surface and block any non-specific binding. So sometimes antibodies, like any protein, will bind to other things if there happens to be something there that matches its binding site. It could even match to the constant domain, the Fc domain of the protein. So you have blocking molecules that prevent that. Then, of course, and then if the antibodies, if your protein of interest is present, when you add the antibody, it will bind. So here's our antibody, and we call this the primary antibody or the first-degree antibody because it directly binds to our protein that we are trying to study. But antibodies themselves, you can't visualize. So the whole point here is to be able to detect the presence of your molecule. So you have to have some way of reporting that your protein of interest is there and that your primary antibody bound to it. There are multiple ways to deal with this. You can tag the primary antibody with some sort of marker, and there's lots of different markers. There's fluorescent proteins or radioactive isotopes you can use. But one really common way is to use another antibody called the secondary antibody. So the secondary antibody will be an antibody that uses the first antibody as its antigen. So again, if let's say you made your primary antibody in a rabbit. So that means you injected your protein of interest into a rabbit, and then you extracted its blood and used it to as the primary antibody. Then you would take rabbit antibodies, and it could be any antibody. In fact, what you usually just do is inject serum from a rabbit, which contains a whole bunch of different proteins, but especially the IgG proteins. Inject that into a different animal. Again, you can't inject it back into a rabbit because the rabbit won't detect it as foreign. But if you inject it into a different species, like say again a goat or a horse or a mouse, and then extract blood from that animal, then you will have secondary antibodies that bind to the constant domain of the first antibody as its antigen. and that protein then can be labeled. And this is usually more efficient because the primary antibodies, because they, usually you're working with limited quantities, so tagging the primary antibody is not very efficient. So it's much more common to tag the secondary antibody. And again, there's lots of different ways to do this. One way is to covalently bind an enzyme. So the enzyme could be, there's lots of different enzymes that can be used this way, but you can covalently attach it to the Fc domain of the secondary antibody. And then you choose an enzyme that has the ability to catalyze a reaction which will produce some sort of colored precipitate as its product. So then you mix together everything. You add your primary antibody to your dish, which has your antigens coated on it. You add your secondary antibody, which will bind to the primary antibody, and the secondary antibody comes pre-attached to an enzyme, and then you add to your dish, your solution, the substrate for that enzyme, or a substrate for that enzyme, that when it reacts with the enzyme becomes some sort of colored substance or precipitate, or becomes visible in some way. And then if your primary antibody binds to your antigen, your protein of interest, then your solution will change color. One common approach to this is called an ELISA, which stands for enzyme. So ELISA stands for enzyme-linked immunosorbent assay, and it looks like this. So you have a dish with a bunch of little culture wells. Each one has been coated with a different amount of your sample solution that you're trying to identify. You applied the primary antibody and then washed off the excess. That's the important part, because of course if you have non-bound antibodies, you want those to leave. Then you apply the secondary antibody, wash that, and then apply a reaction that turns colors if the secondary antibody is present. And so all these yellow wells represent wells that contain your protein of interest, and you can even calculate or estimate the amount of your protein of interest or molecule of interest that's present in each well based on the color intensity. You can use spectroscopy to figure that out. You can also combine these with STS gels. So we've talked about STS gels before. This is a way of separating proteins by size, but of course an STS gel by itself, usually after you do an STS gel, you apply some sort of just wide range protein dye. So all of the proteins in your mixture that are on your gel will show up. So it doesn't matter what kind of protein they are, they're all going to pick up this stain. And if you know the protein size, you can know that your protein is there based on its position. But if you want to know if a specific protein is present on your gel, you can use antibodies. So this process is called an immunoblot, sometimes called a Western blot. And it's done after the gel has been run. You apply the gel to a piece of paper, a fancy piece of paper, or what's called blotting, and essentially the proteins then just leave the gel and get stuck to this paper just through capillary action. And then you take that piece of paper, incubate it with, again, a primary antibody against the protein or the molecule you're looking for, a secondary antibody which binds to the primary antibody, and then some sort of visible marker. Again, it can be an enzyme. There are, in the old days, this used to be done with radioactivity, but in any case, you will then see a band, hopefully, that corresponds to just your protein. So all the other proteins, that might be in the mixture, so all this stuff here, which is, again, sort of proteins you're not interested in, will be invisible because they don't bind to the primary antibody. You can also even, again, use this to estimate the amount of protein. By just the intensity of these bands, you can estimate how much protein is actually there. Another really useful thing to do with antibodies is called immunohistochemistry, or sometimes immunocytochemistry. This involves using antibodies to find proteins or other molecules in tissues or cells. So this is actually a picture that I took of a piece of mouse brain, a very thin slice of mouse brain. And so what I did here is I put actually two different antibodies. One antibody that then had a secondary antibody that had a red fluorescent tag on it, and a different antibody who had a secondary antibody with a green fluorescent tag. So you see both green and red cells here. Only the red cells, though, because they have red and green both, appear yellow. But those cells contain both of the proteins that I was trying to find. So they both of the primary antibodies bound to those cells, but not to the green cells. So again, it's a way to find proteins or to find types of cells, because what I was here interested in is the types of cells here that are colored yellow. They happen to be dopamine neurons in the rat brain. And so this is just my way of trying to find that out. This is back from my old days in graduate school. Now, the last example of protein interaction that we'll talk about, which is a little more dynamic, is muscle protein. So muscle proteins, you probably know, a muscle is basically just a bundle of these fibers, and each cell is one really long fiber, and each fiber is then packed with more bundles of these proteins, and they're surrounded by a complex network of capillaries and sarcoplasmic reticulum, but each protein bundle is one big long fiber called a myofibril. So one muscle fiber is really thousands and thousands of these myofibrils, which is just a protein complex. And we'll talk about the structure of it. But if you look at a myofibril up close, it has this kind of striped appearance to it. And in fact, sometimes skeletal muscle is also called striated muscle, which just means striped or layered. And it's called that because, again, these bands exist within the structure. The bands are separated, or the boundaries of each one are these structures called Z discs. So this is a Z disc, and this is a Z disc, and so each segment of that between those Z discs, actually, what includes the middle of the Z disc, which is the middle of this region called an I band, and then in between each Z disc, you have this bigger region called an A band. But those together, so from one Z disc to another, we call this a sarcomere. And that's all a myofibril is. It's a bunch of these sarcomeres attached end to end. And each sarcomere is a bundle of proteins. And when a muscle is relaxed, the sarcomere is long and the Z discs are far apart. But when the muscle contracts, those I bands get shorter, the Z discs get closer together, and the proteins that make up those bands kind of slide past each other. And that's how muscles work. So that pulling, so the Z discs being pulled toward each other is what causes muscle contraction, and that then applies a force that we use to move. So when you're lifting something off the ground, what you're doing is your arm muscles are contracting by, again, pulling these proteins toward each other, and then creating a force that way. And so the way this works at the molecular level is the sarcomere consists of these bundles of proteins that interact. One is made of this protein called myosin, and myosin proteins are these sort of candy cane shaped structures that sort of wrap around each other. And there's 2 subunits per myosin protein, and the tails, which correspond to the carboxy terminus, are all sort of attached to each other at one end, and then the heads are kind of sticking out the other way, which corresponds to the amino terminus. And so one sarcomere contains a whole bunch of these myosin proteins all kind of wrapped around each other, but pointed in opposite directions. So one bundle facing away from the center, or with the heads facing away from the center, and the other bundle facing the other way. And then the other major protein component of the sarcomere are these things called actin filaments. So actin is a common protein in the cytoskeleton, but it's also important in muscle. An actin filament is really a long chain of these subunits called G-actin. So G-actin just means globular actin. F-actin just means filamentous actin, and it just means that the actin filaments are all bundled together into a long chain. And the interaction between the two happens between the head of the myosin and each sort of alternating segment of the actin filament. So the myosin head has a binding site that fits right in between alternating segments of that actin filament. So again, a sarcomere is two bundles of these myosin proteins facing in opposite directions, and then two groups of these actin filaments that are attached at the Z disc. So again, this is one Z disc, and this is the other. And the actin filaments are attached permanently to the Z discs, but they're not attached permanently to the myosin heads. Also, the actin filaments are sometimes called the thin filaments, and then the myosin bundles together, we call them the thick filaments. So the thin filaments and thick filaments, when at rest, are not attached to each other. So that means that they can slide past each other, which is important because if a muscle is not contracting, you need it to be able to be flexible. So that's an important feature. But when the muscle contracts, these myosin heads start grabbing the actin filaments and they start rotating backwards. So each one kind of rotates backwards and then releases. And then as it does so, as with each sweep, it pulls the thin filament it's attached to towards the center of the sarcomere. And that's happening at both ends. So both ends of the sarcomere are pulling towards the middle. And so that causes the whole sarcomere to contract. And of course, you have a bunch of sarcomeres all working together. And then one myofibril, again, consists of a chain of these sarcomeres all attached end to end. One muscle fiber consists of thousands of myofibrils. And then each muscle contains many bundles of muscle fibers. So all of these proteins working together at the same time create the force from the muscle. So of course, this only happens when the muscle is contracting. When the muscle is relaxed, those proteins should be able to slide past each other, which means you don't want the actin and myosin to attach to each other. So when at rest, there's this other protein complex called tropomyosin, and its partners troponin C and troponin T, and they sit on top of the actin protein, and they just block the interaction between actin and myosin. So the myosin head has a binding site for the actin subunits, but troponin and tropomyosin get in the way. They block the two from interacting. And that's true as long as the muscle's at rest, but when the muscle needs to contract through another complicated series of interactions we don't need to get into, calcium is released into the cell. So calcium ions normally are at a very low concentration in the cytoplasm of most cells, but when a muscle is electrically active, the electrical activity in the muscle triggers calcium channels to open. These are protein channels in the membrane that just let calcium in and raise the calcium concentration. Calcium ions bind to troponin, specifically to the troponin C domain, and that binding causes, again, through induced fit, causes a slight conformational change in the troponin C protein, which then, again, through allosteric effects, leads to changes in the tropomyosin conformation. And again, and all it does is it just shifts slightly out of the way so that the actin and myosin can bind to each other. And that then triggers or allows the cycle that causes muscle contraction to occur. So at the beginning of the sort of muscle contraction cycle, again, up here we've got our myosin head, one myosin head, which again is one of many in this sarcomere, in this thick filament, attached to one of the actin filaments, one subunit of the actin filament. So as long as ATP is present, and that's important because ATP energy is needed for this process. So the myosin head has both an actin binding site and an ATP binding site. But when myosin head binds to ATP, that causes a conformational change in the myosin head that actually reduces its affinity for myosin. So it basically lets go of the myosin head. Pretty much right after that, the myosin head hydrolyzes ATP. So it actually turns out that myosin is also an enzyme. It's an ATPase because it breaks down ATP. And in so doing, the myosin head uses the energy from that hydrolysis to rotate forward. So the myosin head rotates this direction as it hydrolyzes ATP. But it still has ADP. So of course, when we hydrolyze ATP, we are releasing ADP and an inorganic phosphate. That's what the little PI means. So now the myosin head has an ADP and an inorganic phosphate attached. But then it turns out the myosin head has a low affinity for that inorganic phosphate, so it releases it. And that then causes another conformational change along with eventually the release of ADP, in which now that the inorganic phosphate is free, that actually reforms the binding site from actin and myosin. So myosin then rebinds to actin, but this time at a different position, because initially it was attached to this actin subunit, but because the head rotated, now it's in position to bind to this actin subunit. And then when ADP is released, that causes the myosin head to rotate again, but this time this direction. So it rotates essentially back to its original shape. But we call this the power stroke, because during this rotation, again, because it's firmly attached to that second actin subunit, it rotates, but it also creates a force. So essentially, what happens is the myosin head pulls the actin filament this way, or you kind of have both things happening at the same time. The thick filament is pulled that way, and again, this would be pulling. So presumably somewhere down here, you would have your Z disc. And then down at the other end, you would have another Z disc, and with another set of thin filaments attached to it. And then this myosin head would be attached to it at that end, and it's doing the same thing in the opposite direction. It's pulling the other Z disc again toward the middle, and that creates the force. And then after that, the myosin head has the ability to bind to another molecule of ATP. So the whole cycle just starts over again. So now another ATP can bind to myosin, and the whole thing repeats. And like I said, this will continue as long as calcium's present. So calcium has to be present in order to prevent tropomyosin from getting in the way. If calcium concentration goes down, tropomyosin moves back into place, and myosin and actin won't be able to bind. And that happens when you stop contracting that muscle. So when the muscle is relaxing, then there's no calcium, and the proteins can slide past each other. The other thing that will stop this cycle, though, is when ATP runs out. So this will continue as long as ATP is present, but if ATP is depleted, meaning the cell or the muscle fiber has depleted all of its energy, or if it's depleted of oxygen. Later on, we'll talk about, in a later chapter, we'll talk about how ATP is made, why oxygen is necessary. So if a muscle runs out of oxygen or runs out of fuel to make ATP, then ATP concentrations will go down, and this process won't be able to continue. But importantly, the place where it will get stuck is right here. So without ATP, the myosin head and the actin filament will be attached, because that's what ATP does. The ATP doesn't actually directly drive the muscle. ATP's job is to trigger the release of the myosin head from the actin filament. And this is why, in fact, when a muscle is depleted of oxygen or of ATP, the muscle essentially freezes, because, again, if myosin and actin are bound to each other, then the muscles, the thick filaments and the thin filaments, can't slide past each other. They're just sort of locked in place. And in fact, this is what happens when you die. So when an animal dies, the muscle tissue very quickly uses up all of its oxygen, and then the myosin heads become locked to the actin filaments. And this means that the muscle essentially will not be able to move freely. And so this is why when animals die, their bodies become stiff. This is the process of rigor mortis. Same thing happens though if you are really overusing a muscle and you use up all the oxygen in the muscle too quickly. Under certain circumstances, you can have a muscle cramp. So muscle cramps can also be caused by lack of oxygen to the muscle, partly for this reason as well. So that is it for this chapter. Again, we were just going through some examples of protein interaction. Again, you could use any type of protein you want to use as an example. In the next chapter, we'll deal specifically with a special kind of protein called enzymes, and because they are especially important for cell function as well. All right, that's all.